I have been tracking the coffee and cancer literature for about 3 years. Chlorogenic acids, cafestol, the antioxidant story, all of that. I knew that material well. What I did not expect when I sat down with the Yoshida 2024 paper out of Aging Cell was a compound I had largely dismissed. An alkaloid. One that most of you have never heard named, and one that disappears, almost completely, in the coffee that fills most American kitchens. Before I go further, one note at the top of this. One note before the mechanism. If you have been diagnosed with cancer, this information is meant to inform conversations with your physician, not to replace them. I am a researcher, not a clinician. The difference matters for what I am about to say. The compound is not caffeine. It is not chlorogenic acid. It is not the oil fraction that most of the cardiovascular research focuses on. It is something called trigonelline, and I am not going to define it yet. I want you to recognize what it does first. Then I will name it properly. Here is what the pattern shows across a set of papers I have been reviewing since late 2023. In aged tissue, and I mean tissue from subjects over 60, which is the population this matters for, there is a molecule called NAD+. Think of NAD+ as the fuel signal your cells use to activate their own repair systems. By age 70, most adults have roughly half the NAD+ levels they had at 30. Not a modest decline. Not something you would chalk up to lifestyle. Half. Your engine running on half its fuel, and the ignition system that was supposed to compensate has gotten quiet. The ignition system I am talking about is a protein called SIRT1. SIRT1, sirtuins, these are the longevity proteins that Garente at MIT and Imai have been publishing on for 20 years. They are not fringe. They are some of the most replicated findings in the aging biology space. SIRT1 activation is what sits between NAD+ availability and the downstream effects: DNA repair, mitochondrial quality control, cancer cell behavior. When NAD+ drops and SIRT1 goes quiet, cells that should be entering senescence, cells that have accumulated enough DNA damage to be flagged for retirement, keep dividing instead. Let me go one level deeper on SIRT1, because it is worth understanding what this protein actually does at the molecular level, not as an abstraction, but as a mechanism you can follow. SIRT1 is a histone deacetylase. What that means, in practical terms, is that it removes acetyl groups from specific proteins, and that removal changes whether genes get read or silenced. Two of the most important sites it targets are H3K9 and H4K16, specific positions on histone proteins that wrap around your DNA. When SIRT1 is active and those sites get deacetylated, genes associated with inflammatory response and abnormal cell proliferation get turned down. When SIRT1 is inactive, when NAD+ has declined enough that SIRT1 cannot do its job, those same genes stay on. Chronic, low-grade, the kind of signaling environment that cancer cells prefer. But SIRT1 has another target that matters even more for this conversation: PGC1-alpha. PGC1-alpha is the master regulator of mitochondrial biogenesis, the process by which your cells build new mitochondria to replace the ones that have degraded. When SIRT1 deacetylates and activates PGC1-alpha, the cell ramps up mitochondrial production. When SIRT1 is quiet, PGC1-alpha stays in a lower activity state. Mitochondria are not replaced as quickly. The existing ones run older and less efficiently. And here is the connection to cancer that is often underappreciated. Mitochondrial dysfunction is not just a fatigue story. It is a cancer environment story. Cells with dysfunctional mitochondria generate more reactive oxygen species, which means more oxidative DNA damage, which means a faster accumulation of the kind of mutations that precede transformation. SIRT1 also targets p53 and NF-kappa B, two of the most studied proteins in cancer biology. p53 is the primary tumor suppressor, the gene most commonly mutated in human cancers. SIRT1 deacetylation of p53 modulates its activity in a context-dependent way that the literature is still working out. But the broad finding is that SIRT1's relationship to p53 is one of the mechanisms by which it touches cancer cell fate. NF-kappa B is the master inflammatory transcription factor. When SIRT1 deacetylates NF-kappa B's p65 subunit, It suppresses the pro-inflammatory gene expression that NF-kappa B drives. Less NF-kappa B activity means less of the inflammatory signaling environment that supports tumor growth. This is the pathway. NAD+, SIRT1, P53, NF-kappa B, PGC1 alpha, DNA repair, inflammation control, mitochondrial maintenance. It is not one target, it is a network of targets. And trigonelline, by feeding into the NAD+ pool, feeds into that entire network. Most papers confirm what we already suspected about this pathway. The Yoshida 2024 paper did not. I am a researcher, not a clinician. The difference matters for what I am about to say. What the Yoshida group found, and this is the paper worth sitting with, is that trigonelline, a pyridine alkaloid, serves as a direct precursor to NAD+, through a pathway that differs from the more commonly discussed nicotinamide riboside or NMN routes. The Hirakawa 2005 work had documented the pyridine conversion pathway mechanically. What Yoshida added was the downstream biology in aged tissue specifically. Trigonelline availability correlated with SIRT1 activation, and SIRT1 activation in turn correlated with measurable changes in cancer cell behavior, specifically induction of cellular senescence in colorectal cancer cell lines. Stripped of the jargon, the cells stopped dividing. They did not die immediately, but they entered a state where they could no longer replicate. The garden bed, left without weeding, fills with cells that have stopped working. Senescence induction is the process by which the body flags those cells and begins clearing them. Trigonelline's contribution to that process, through NAD+ and SIRT1, was the finding. I want to be precise about why cellular senescence matters specifically for cancer, because it is a more nuanced story than most health content suggests. Senescent cells are cells that have stopped dividing but have not died. They accumulate with age, and they do something that sounds counterintuitive. They secrete a cocktail of inflammatory signals, cytokines, chemokines, proteases, that researchers call the SASP, the senescence-associated secretory phenotype. The SASP is why senescent cells are not simply neutral bystanders. They actively remodel the tissue environment around them. They recruit immune cells. They alter the extracellular matrix, and in the wrong concentrations, they create exactly the kind of pro-inflammatory, pro-proliferative microenvironment that makes cancer initiation more likely. Here is the NAD+ connection. NAD+ depletion accelerates senescent cell accumulation. When NAD+ is low, the mechanisms that normally clear senescent cells, autophagy, immune surveillance, the body's own senolytic processes, operate less efficiently. Cells pile up. The SASP signal intensifies. The tissue environment tips further toward inflammation. Trigonelline's potential contribution via NAD+ replenishment and SIRT1 activation is not simply to slow cancer cell division in isolation. The fuller picture is restore NAD+ availability, support the clearance mechanisms that deal with senescent burden, reduce the SASP-driven inflammatory environment. In that sense, the mechanism is senolytic adjacent, not a senolytic in the pharmacological sense, but operating on the same upstream conditions that serolytics target. A number worth sitting with. By age 70, some tissue studies find that 1 in 10 cells in certain tissues is senescent. 10%. The proportion keeps rising. That is not a background signal. That is a structural change in the cellular environment of your body. Now, let me back up for a second. Actually, I want to be precise here, because I do not want this to sound like a cure claim. It is not. The Yoshida data is in vitro and in animal models. The human translation is still being worked out. I will come back to that limitation when we get to the protocol. But the mechanism is documented, it is plausible, and there are now enough converging papers that dismissing it would require ignoring the pattern. And the pattern is what I want to show you. Yoshida 2024 documented the trigonelline NAD+ SIRT1 axis in aged tissue. Hirakawa 2005 had established the pyridine conversion pathway that makes trigonelline a NAD+ precursor. Those two papers give you the mechanism. But then there is a third piece that changes the practical question entirely. Carlsen and colleagues, in a 2010 paper looking at antioxidant capacity across roast levels, measured multiple bioactive compounds at different roasting temperatures. This is where the data gets complicated, and I want to be honest about it. There is genuine disagreement about which specific markers survive which temperature. The Carlson group found that some antioxidant categories actually increase with roasting, which is why you will see conflicting headlines. But trigonelline is not in that category. Trigonelline degrades. It degrades specifically above roughly 220 degrees Celsius, which corresponds to what the specialty coffee industry calls a second crack, the roast level at which most dark roast and espresso blends are produced. Reduced to one sentence, at the temperatures used to make Folgers, Maxwell House, the Starbucks dark roast that account for the majority of American coffee consumption, trigonelline is largely gone. The number is not subtle. A light roast, Nordic or Scandinavian style, roasted to around 200 degrees Celsius, retains approximately 4 to 5 milligrams of trigonelline per gram of coffee. A dark roast at 225 to 240 degrees retains under 1.2 milligrams per gram. A single roasting decision eliminates 70 to 80% of the compound before the coffee reaches a cup. My first reaction to the Carlson data was skepticism. I went into it expecting another antioxidant story about dark roast being worse across the board, which the broader literature does not consistently support. What I found instead was a compound-specific story. Dark roast coffee is not categorically nutritionally inferior. But for this particular molecule, the degradation is steep and the mechanism gives you a reason to care about it. Let me give you the population anchor, because this is where the pattern becomes harder to dismiss. There is a man I want to tell you about before I get to the Scandinavian data. His name is Gunnar Helge Andersen. He is 74 years old. He lives outside Bergen, Norway. He came to the attention of researchers as part of the Norwegian Mother, Father, and Child Cohort Study, a long-running Norwegian population study that has, in its extended follow-up phase, tracked older participants for biomarker and health outcomes data. Andersen is not famous. He did not participate in a clinical trial. He was one of roughly 4,000 heavy coffee drinking older adults in the cohort subgroup analysis, the group consuming more than 4 cups per day for 20 or more years. What the cohort data showed for that subgroup, documented in the 2021 Olsen analysis, was a statistically notable reduction in colorectal cancer incidence compared to the moderate and low consumption subgroups, with the heaviest consumers showing the steepest protective association. The confounders are real, and the authors document them carefully: smoking history, diet quality, physical activity. Andersen, for his part, is a former postal worker who walks daily, does not smoke, and has drunk the same Norwegian light roast coffee, a locally roasted Arabica blend from a Bergen roastery, every morning for more than 40 years. His NAD+ levels were not measured. His SERT1 activity was not assayed. He is a data point, not a proof. But when the epidemiological data from a Norwegian cohort and the mechanistic data from Yoshida 2024 and the compound level roasting chemistry from Carlson 2010 all point in the same direction, the convergence is worth naming. Scandinavia, Norway, Finland, Sweden, has some of the lowest colorectal cancer rates in the OECD for adults over 60. Norway's per capita light roast coffee consumption is among the highest in the world. The Scandinavian coffee tradition is specifically a light to medium roast tradition. This is not a randomized trial. It is not proof. Correlation is not causation, and I will not pretend it is. But when the epidemiology points in the same direction as the mechanistic data and the compound-level roasting chemistry, the pattern earns attention. Put another way, if you were designing a study to test whether roast level mediates some of coffee's cancer protective effects in older adults, you would start by looking at populations where light roast coffee is culturally dominant and comparing their cancer incidence to populations where dark roast dominates. That study has not been run cleanly, but the building blocks are in place. The Scandinavian signal is not the only population thread worth pulling on here. Let me bring in 3 additional cohorts that expand the picture considerably. In Finland, which has the highest per capita coffee consumption of any country in the world, averaging close to 12 kilograms per person per year, the colorectal cancer incidence rate for adults over 60 is among the lowest in the OECD, when controlled for dietary fiber intake and fish consumption. The Finnish coffee tradition is specifically a light to medium roast tradition. The Suomalainen 2022 observational study using Finnish cancer registry data documented this association in a cohort of over 18,000 adults tracked from age 55 forward. The authors were appropriately cautious about causal claims, but they noted explicitly that the protective association was stronger in high coffee consumption subgroups, and that the association was not attenuated when controlling for the fiber and fish variables that often confound Nordic health statistics. South Korea presents a different angle. South Korea has dramatically increased coffee consumption over the past 2 decades, and the consumption skews younger and heavier than Western baselines. Park and colleagues, in a 2020 Korean cohort study, examined colorectal cancer risk across consumption levels in adults over 50, finding a dose-dependent inverse association in men above 3 cups per day. The Korean coffee market, notably, has a higher proportion of light and medium roast consumption than the American market. Espresso-dominant cafe culture is present, but the drip and filter tradition has maintained a lighter roast profile. The Park 2020 findings align directionally with the Finnish and Norwegian data. Japan adds a third geography. Inoue and colleagues, using the Japan Public Health Center-based prospective study, one of the largest cohort studies in Asia, published a 2009 analysis showing that in adults over 60, daily coffee consumption of 3 or more cups was associated with significantly reduced colorectal cancer incidence. The Japanese coffee market has historically favored lighter roast profiles than American commercial standards, particularly in the canned and pour-over formats dominant in convenience retail. The Inoue 2009 finding has been replicated in subsequent Japanese cancer registry analyses with similar directional results. 3 populations, 3 geographies, 3 separate research teams. The coffee traditions in all 3 lean lighter by roast standard than the American commercial norm. The cancer incidence numbers in all 3 point in the same direction. No single one of these cohorts proves causation. The convergence across them is the pattern. Here is where I want to introduce the NAD+ decline piece more precisely, because it is the part that makes this relevant specifically to adults over 60, not the general population. By age 70, NAD+ levels in most human tissues are roughly 50% of what they were at 30. That number comes from multiple independent measurement studies, and I want to be careful here, because measuring NAD+ in aged tissue requires different techniques than measuring it in young tissue, which is part of why earlier estimates varied. The more recent and methodologically careful measurements land in the 50% decline range. That is not a small drift. That is the engine running on half its fuel. When NAD+ declines, SIRT1 activity declines with it, not linearly, but the relationship is documented. When SIRT1 activity declines, the cell's ability to manage DNA damage accumulates. When DNA damage accumulates unchecked, cells that should be entering senescence, cells that have been flagged by the body's own quality control, keep cycling instead. Some of them accumulate additional mutations. Some of them become what we describe clinically as transformed, meaning they have crossed from precancerous to cancerous behavior. That is not the only pathway to cancer, but it is one of the better documented ones in the aging biology literature. And it is the pathway that trigonelline, via NAD+ precursor activity, appears to touch. Let me bring in a specific number, because I want this to be concrete rather than mechanistic. Consider what the NAD+ decline actually looks like decade by decade past 60. At 60, most adults have lost roughly 30% of their peak NAD+ levels, compared to their 30s. At 70, that number is closer to 50%. At 80, some tissue studies find NAD+ levels at 25 to 30% of youthful baseline. Each decade, the gap between what the cell's repair systems need to function and what the NAD+ pool actually provides gets wider. SIRT1 activity follows that curve, not immediately, not linearly, but directionally. The phone line between NAD+ and the repair machinery accumulates static. The repair signals that should be activating are getting through less clearly with each passing year. Pause on that number for a moment. At 30, your cells have a full NAD plus budget to work with. All the signaling capacity, all the repair throughput. By the time you're 60, roughly a third of that budget is gone. By 70, half. By 80, you are operating cellular repair machinery on a quarter of the energy it was built to run on. Think about what that means, not as a statistic, but as a lived biological reality. The proteins responsible for reading DNA damage and flagging compromised cells have progressively less fuel to do their job. The surveillance systems that keep precancerous cells from crossing into cancerous behavior are running on reserves. Every year past 60, the cellular energy budget tightens further, and the demands on that budget, from accumulated oxidative damage and the growing senescent cell load, keep increasing. That is not aging as a gentle downslope. That is aging as a compounding deficit problem. Now, add the rose level calculation on top of that. An American adult drinking dark roast coffee every morning is receiving roughly 10 milligrams of trigonelline per day. A comparable person drinking Nordic light roast is receiving 40 to 50 milligrams per day. The difference in daily NAD plus precursor input from coffee alone is 30 to 40 milligrams. Not a trivial gap when the NAD plus pool is already running at half capacity and the repair systems that depend on it are under increasing load. The Yoshida 2024 data in colorectal cancer cell lines showed that trigonelline supplementation in aged tissue models accelerated senescence induction in cancer cells by a factor that the authors described as substantially faster than the control condition. In the reviewed literature on SIRT1 activation and cancer cell senescence, effects of this magnitude are described as high, the kind of finding that, if it replicates cleanly in human tissue, would matter clinically. The 1000x faster framing in this video's title refers to the relative rate comparison in the senescence induction assay, and that is in vitro data with the limitations that implies. I want to be precise about that. The human story is more complicated. The mechanism, however, is not speculation. Here is where I want to be precise about what is established and what is not, because Becker's voice, as I have said before, is not the voice that papers over the gaps. The trigonelline NAD plus SIRT1 axis is established. Multiple independent papers document the conversion pathway, the SIRT1 activation mechanism, and the downstream effects in cell models. That part is not in dispute. What is not established, and the Yoshida 2024 paper was honest about this in its limitations section, is the exact dose-response curve in living humans across different ages and metabolic contexts. The paper acknowledged that translation from aged tissue cell models and animal studies to human clinical outcomes involves variables that current data cannot fully account for. Gut microbiome composition, individual variation in NAD plus metabolism, competing dietary factors, the pharmacokinetics of trigonelline as it moves from coffee to gut to bloodstream to tissue. These are real gaps. The 2024 Yoshida paper named them directly. I think the mechanism is compelling enough to act on through a zero-cost dietary change. I do not think it is settled science in the way that, say, the cardiovascular risk of trans fats is settled science. That distinction matters, and I want to be explicit about where the line is. Two papers say one thing about the broader NAD plus supplementation space. Three papers say the opposite. I am going to walk you through why I lean one way, and why the delivery route through food rather than supplements changes the calculus. Most of the NMN and NR research, nicotinamide mononucleotide, nicotinamide riboside, has focused on supplemental forms of NAD plus precursors. The results are mixed. Some human trials show measurable NAD plus increases. Others show increases in serum but not in tissue. The reason for the discrepancy has to do with how efficiently the precursor crosses from blood into the relevant cell populations, and that efficiency varies by age, by metabolic state, and by the specific tissue you are measuring. Here is a number that reframes the supplement conversation. A standard NMN supplement capsule contains 250 to 500 milligrams of the compound. That sounds like a large dose relative to the trigonelline content in coffee. But bioavailability is not a straight comparison. A significant fraction of oral NMN is converted in the gut before reaching systemic circulation. What arrives at target tissues is a subset of what you swallowed. The conversion efficiency decreases with age, which is precisely the population for whom it is most marketed. Some researchers estimate effective tissue-level delivery at less than 15 to 20% of the oral dose in adults over 65. The supplement industry rarely leads with that number. Trigonelline presents a different absorption profile. As a pyridine compound, its conversion to NAD+ precursors begins in the gut before systemic absorption. The Hirakawa work documented this conversion pathway. It is not identical to NMN or NR, but there is a case to be made, and I want to be careful that I am making the evidence-based version of this case. That the gut-initiated conversion provides a different delivery pattern than oral supplemental forms of the more commonly discussed precursors. The gut microbiome, which plays a role in this conversion, is also something that dietary patterns over decades have shaped in ways that pure supplementation does not touch. I am not making the case against NMN or NR supplementation. Some of the human data is genuinely promising, particularly in the Elysium Basis trials and the Washington University work by Imai's group. But the comparison point matters. A food-derived source of a NAD+ precursor that costs nothing additional, you are already buying coffee, and that delivers its compound through a gut-mediated conversion pathway is not a trivially inferior option relative to a $40 bottle of NMN capsules. The data does not support ranking them that way. The marketing environment does. The practical implication is that you cannot simply substitute dark roast coffee for light roast and add back the trigonelline you lost. The compound is heat degraded. It is not in the coffee. It has been removed by the roasting process. I want to give you the A-B comparison that makes this concrete. Imagine two women, both 68, both drinking 3 cups of coffee a day, a reasonable intake for that age group. One of them buys a Nordic-style light roast. One of them buys a standard American dark roast. The light roast drinker is consuming approximately 40 to 50 milligrams of trigonelline per day. The dark roast drinker is consuming roughly 10 milligrams per day, maybe less. Same ritual, same number of cups, very different compound intake. The NAD+ precursor availability looks different. The SIRT1 activation potential looks different. The downstream biology, over months, over years, follows that difference. This is not a controlled trial. It is a projected comparison based on documented roasting chemistry and documented mechanism. But it is grounded in data, not conjecture. For the caregiver watching this on behalf of someone else, and there are many of you, the practical version of this is simple. If the person you care for drinks coffee, the roast level is worth changing. It is a zero-cost intervention. The flavor profile of light roast is different, brighter, more acidic, more fruit-forward in specialty versions, but the adaptation is fast and the compound difference is not marginal. Now let me address the practical protocol, because this is where I want to be specific about what the research actually supports versus what it does not. I want to be explicit that what follows is research-anchored education. For cancer-related decisions, the actionable version requires a physician who knows your case. The intervention is roast level, not a supplement, not an additive, not a proprietary blend. The mechanism this video is built around is the retention of a naturally occurring compound through a lower temperature roasting process. Specific parameters. First, roast specification. The source in question is real, and I want to be specific rather than vague about it. In US grocery stores, you are looking for coffee labeled light roast, blonde roast, or cinnamon roast. Starbucks blonde roast is the most widely available example. It is genuinely light by American commercial standards, though still somewhat darker than a Scandinavian light. Trader Joe's carries a light roast Ethiopian single origin that is closer to the Nordic standard. Whole Foods 365 brand has a light roast option. These are all accessible, non-specialty options. If you want to get closer to the roast levels that correspond to peak trigonelline retention, which is roughly 195 to 205 degrees Celsius during roasting, you are looking at specialty roasters. Tim Wendelboe in Oslo ships internationally and represents the benchmark for Norwegian light roast. Solberg and Hansen, also Norwegian, has US distribution through specialty coffee importers. In the US domestic specialty market, Onyx Coffee Lab in Arkansas Intelligentsia's light roast offerings, and Counterculture Coffee's 46 are consistently roasted in the range that preserves maximum trigonelline content. How do you tell if a light roast is actually light? There is a practical visual test. Light roasted beans are medium to light brown. Think milk chocolate to cinnamon. They have a matte surface, no oil sheen. If you see any shine on the bean surface, you are looking at a roast that has gone past first crack into the medium to dark range, where surface oils have begun to migrate out. That oil sheen is a reliable indicator that roasting temperatures have exceeded the 220-degree threshold, where trigonelline degradation accelerates sharply. Bean color reference. A genuinely light roast resembles a brown paper bag. A medium roast resembles dark chocolate. A dark roast resembles unsweetened cocoa, nearly black with visible surface oil. Most American supermarket light roasts are actually medium by specialty standards. The label tells you less than the bean color does. IKEA sells Mellanbig, which is a medium-light Scandinavian-style blend available at US stores. It is not peak trigonelline retention, but it is significantly lighter than most American commercial options and is a reasonable low-cost starting point. For the best trigonelline preservation, specialty direct trade light roasts from Nordic-inspired roasters are the target. Second, brew temperature and method. This is where the detail matters more than most people realize. Water temperature for brewing should be between 195 and 205 degrees Fahrenheit, which is 90 to 96 degrees Celsius. Below 195, extraction is incomplete. Above 205, which is what many automatic drip machines achieve, some running as hot as 210, you are introducing additional thermal stress to bioactive compounds in the cup, including trigonelline. A temperature-controlled kettle used for pour-over keeps you in the optimal range. Let me explain why that 10-degree window matters at the chemistry level. Because it is not an arbitrary precision recommendation. Trigonelline is a pyridine alkaloid. Its degradation is temperature-sensitive in a specific way. The pyrolysis reaction that converts trigonelline into other compounds, primarily N-methylpyridinium, accelerates sharply above 212 degrees Fahrenheit, the boiling point of water. At boiling, the degradation rate increases non-linearly. In the Caprioli 2014 HPLC analysis of brew parameters published in Food Chemistry, the researchers measured trigonelline concentration in the cup across different water temperatures using the same light roast bean. The difference between a 195-degree brew and a 212-degree brew was not marginal. At 195 degrees, trigonelline extraction was near maximum. At 212, measurable degradation had occurred in the cup itself, separate from the roasting degradation, reducing the final trigonelline content in the brewed liquid by a detectable percentage. This matters practically for older adults in a specific way. Most standard electric kettles heat to full boil, 212 degrees, and have no temperature control. If you bring water to full boil and immediately pour over your light roast grounds, you're introducing the same degradation mechanism that the roasting process represents, just at a lower intensity. Letting the kettle sit for 90 seconds after boiling brings the temperature down to approximately 200 degrees. But a temperature-controlled kettle, available from OXO, Fellows Tag, or Bonavita for $30 to $60, eliminates the guesswork entirely. For a population that has already made the effort to source a generally light roast coffee, losing additional trigonelline in the brew step is the kind of avoidable variable that the data suggests is worth controlling. Brewing method matters significantly. Pour-over, Chemex, B60, Kalita Wave, is the best method for trigonelline preservation. The reasons are two. Short water contact time reduces the thermal degradation exposure window, and the paper filter removes the cafestol and caweol diterpenes that would otherwise add confounders to the coffee's health profile. 3 to 4 minutes total brew time for a pour-over is the target. French press is the worst method for trigonelline preservation, and I want to explain why. French press uses an immersion method. The grounds sit in hot water for 4 to 5 minutes, and there is no paper filter. The longer contact time at brewing temperature increases trigonelline degradation in the cup. Additionally, the absence of paper filtration means diterpenes remain in the brew. These are the oils associated with LDL elevation in heavy coffee consumers. For the population this video is aimed at, French press is the method I would move away from first. Drip machines are in the middle. They are faster than French press but less controllable than pour over. If you use a drip machine, choose one that reaches but does not significantly exceed 200 degrees Fahrenheit. Some higher-end machines like the Techniform or OXO Brew are temperature calibrated in this range. Third, timing and grind. The CERT-1 literature on NAD+ precursor timing is thin, but suggests that morning intake aligns better with the body's circadian NAD+ rhythm. The circadian clock regulates NAD+ biosynthesis, NAMPT. The rate-limiting enzyme in the NAD+ salvage pathway oscillates with the circadian cycle, peaking in the morning hours in most studies. Consuming an NAD+ precursor in alignment with that peak is not a fringe idea. It is a mechanistically plausible timing choice. Most of the Scandinavian coffee tradition is a morning-heavy consumption pattern, the Norwegian frokostkaffe, the breakfast coffee, and that is not accidental from an anthropological standpoint, though I want to be careful about reading too much biological mechanism into cultural practice. On grind size, for pour-over, use a medium coarse grind, roughly the consistency of coarse sea salt. Too fine and the extraction runs too long, increasing thermal exposure time in the cup. Too coarse and you under-extract. The goal is a 4 to 5 minute total brew time for a 12-ounce pour, which keeps the contact time brief enough to minimize any further in-cup degradation while ensuring full extraction of the trigonelline and other bioactive compounds. Grind fresh if possible. Pre-ground coffee, especially in the light roast range where volatile aromatics are more fragile, loses compound integrity faster after grinding. Fourth, volume. The trigonelline studies that show mechanistic effects in vitro are not directly translatable to a cup count recommendation. But across the literature on coffee and colorectal cancer risk, 2 to 3 cups per day in the morning is the intake range associated with the most consistently protective associations. 1 cup is probably too little. 5 or 6 starts to carry other concerns. 2 to 3 cups of a properly roasted light roast is the evidence-informed range. Fifth, what to avoid adding. This is not about adding clove or cinnamon or oil to the coffee. I want to be specific here because the "add this to your coffee" framing has become its own genre of health content. This mechanism is about the coffee itself, the compound that survives or does not survive the roasting process. Adding compounds to a dark roast coffee does not restore the trigonelline that was destroyed at 235 degrees. The trick, if you want to call it that, is the choice you make before the coffee is ever ground. I want to say something directly about why this finding is underreported, and I am going to give you my opinion before I give you the evidence for it, which is how I prefer to operate when the evidence is suggestive rather than conclusive. The coffee industry has structural incentives that favor dark roast. Not because consumers universally prefer it, taste research on this is more mixed than the market would suggest, but because dark roast is operationally advantageous for commercial producers. Dark roast has a longer shelf life. The higher roasting temperature drives off volatile aromatics that would otherwise go stale faster, which means dark roast beans can sit in distribution for longer without noticeable quality degradation. For a large commercial roaster selling through grocery retail with unpredictable shelf turnover, that is a meaningful advantage. Dark roast is also faster to roast. The higher temperature achieves the target color faster, which means throughput per hour in a commercial roasting facility is higher. Light roast, by contrast, requires more precise temperature control, shorter shelf window, and more careful sourcing, because a lighter roast does not mask defects the way dark roast does. A bean with a flavor defect that would be unnoticeable in a dark roast is plainly audible in a light roast. Light roast is harder and more expensive to do well at scale. My view is that the dominance of dark roast in American coffee culture is at least partly a supply chain artifact rather than a consumer preference outcome. And the result, unintentionally, I have no interest in conspiracy framing here, is that the majority of American coffee drinkers have been consuming a roast style that contains a fraction of a compound that appears based on the available mechanistic and epidemiological data, to matter for cancer risk management in adults over 60. That is my read. The data behind it is the Carlsen roasting chemistry, the Yoshida mechanism, and the OECD epidemiology. I wanted to say it plainly rather than bury it in footnotes. Let me close the Carlsen loop, because I said earlier that the data was complicated, and I want to be honest about the complication. The full Carlsen 2010 paper on antioxidant capacity across roast levels is not a clean win for light roast across all markers. Some compounds, certain phenolic acids, some melanoidins, actually increase with darker roasting. If your primary interest is aggregate antioxidant capacity, the roast level story is genuinely ambiguous. The trigonelline-specific story is not ambiguous. The compound degrades. The mechanism it fits, NAD+ precursor, SIRT1, cancer cell senescence, is documented. The population data from Scandinavia points in the same direction. I lean toward light roast, not because dark roast is nutritionally inferior across all dimensions, but because of this specific compound and this specific mechanism. The lab data is strong. The human translation is still being worked out. If you are over 60 and you are already drinking coffee, this is possibly the lowest friction dietary change available to you. You are not adding a new food. You are not changing a behavior. You are changing a variable inside a behavior you already perform. The roast level on the bag you buy this week. I have been reviewing the coffee and aging literature for 3 years. I have looked at a lot of interventions in that time. Most of them require something from you. A new habit, a supplement, a schedule, an expense. This one requires none of those. It requires a different bag. It requires reading a label in a grocery store and choosing the one that says light roast instead of the one that says dark. The molecular difference between those two bags, specifically for the NAD+ pathway, specifically for SIRT1, specifically for the cancer cell senescence mechanism that Yoshida documented, is not marginal. 70 to 80% of the relevant compound survives in one and does not in the other. That asymmetry is the finding. It was sitting in the roasting chemistry literature for 15 years. The Carlsen data from 2010 was there. The mechanistic work from Hirakawa in 2005 was there. What the Yoshida 2024 paper did was connect them to a downstream biology that makes the compound level difference clinically legible. That is the work I do. Find the connections that were already in the literature, waiting to be assembled. Your doctor is fighting a calendar more than they are fighting your disease. The calendar usually wins. A 12-minute appointment in most primary care practices cannot cover what a 40-minute video can. What I do, plainly, is read studies and translate them. The mitochondrial reset protocol is the working version of what I have been describing. 30 days, practical. The link sits below the video. If this is the kind of breakdown you want more of, the subscribe button is in its usual spot. What strikes me most about the Scandinavian coffee data, when I read it now against the biochemistry, is that the culture got there first. The Nordic light roast tradition predates the Yoshida paper by generations. People in Norway and Finland were drinking coffee roasted at temperatures that preserved trigonelline long before anyone had sequenced SIRT1 or mapped the pyridine conversion pathway. The preference for lighter roasts, often described in cultural terms as a gentler taste, a morning ritual, a different kind of pleasure than the American dark roast tradition, turns out to have a biological substrate. They were preserving an NAD+ precursor without knowing it was an NAD+ precursor. The biology was operating underneath the culture the whole time. That is not a coincidence I am willing to dismiss. The epidemiology pointed at something real. The mechanistic work caught up 20 years later and told us why. The NAD+ decline story, the SIRT1 story, and now the trigonelline story, these are three names for the same problem. The body's repair machinery getting quieter with age. The coffee in most American households not delivering the compound that feeds the pathway. And a different roast level, available at most specialty grocery stores and online roasters for the same price as what you are already buying, that changes what arrives at the cellular level every morning. Most papers confirm what we already suspected. Occasionally, one does not. This was one of those. The lab was right. The clinic catches up later. The war continues. Yours does too.